Hello and welcome to this video on the phospholipid bilayer that makes up the membrane of all your cells. To understand what the lipid bilayer is and why it is important, let's imagine what would happen with a minimal or simple cell membrane. That is something that only holds the cell content inside the cell, or an approximation thereof. With this theoretical cell membrane, the content inside the cell, such as salt or electrolytes, would freely move in and out via diffusion. The same thing would apply to water. This would lead to mass movements of both electrolytes and water at any given time. In the best case scenario, it would cause cell disruption, but the cell could live for the most part. It would certainly struggle, but it would manage. In the worst case scenario, it would cause too much water to enter or leave, leading to cell death through lysis or collapse. So yes, it would work, but that simplicity comes at the cost of function and cell health. Given that your body doesn't experience this, obviously something more is going on. And that's why we now come to why the simplicity of the membrane isn't good enough, but the human cell membrane isn't a minimal cell membrane. The phospholipid bilayer membrane can be largely understood from its name. It's two layers. These two layers are made from a matched pair of structures. The structures are a phosphate head, often depicted as a spherical head, and from the head hang two tails. These are made of lipids or fats. When the matched pairs are put in water, as part of the membrane, they behave in a specific way. Lipid or fat tails are hide from the water inside the membrane. The phosphate head sticks out, facing the world outside the cell that is full of water, and inside the cell, which is generally also full of water. This phenomenon is a result of the way lipids are repelled by water. It's also why oil and water do not mix. The hydrophilic area, or the area that doesn't like water of the phospholipids, form hydrogen bonds, and when they're with water, they tend to try and hide from it. Because there is a degree of polarity, that is a certain amount of attraction and repulsion like magnets, it creates this inside and outside aspect to it. By contrast and relative comparison, the phosphate heads are hydrophilic or water-loving. This is why they orient themselves outwards, towards the water. The end result of this initial description can be compared to a sandwich. A top layer of bread or phosphate heads, a filling of lipid tails, followed by a bottom layer of bread made up of phosphate heads. The end result or effect of this initial information is significant. It's the combination of lipids and phosphate heads that make it hard for electrolytes to diffuse through, but still possible if a certain gradient exists, but it's much more difficult. The other effect is much larger. That is based on the charge, size, and solubility of what can pass through the phospholipid bilayer. Large things can be all sorts of things, for example, proteins, big chemicals, and other cells such as bacteria. Charged things include hormones, lipids, salts, and more. Insoluble is somewhat more difficult to explain. Because the inner part of the membrane is made up of lipids or fats, only something that is soluble in lipids can easily pass through. This means that water-soluble things can't get through the membrane very easily, if at all. If it's lipid-soluble, it can, and this is particularly important when we're talking about things like drugs, but that's beyond the scope of this video. Just be aware of it. So now we have the problem of how do we get things through the membrane if they don't fit those three criteria of size, charge, and lipid solubility? The answer is transporters. There are different kinds of transporters. We can broadly describe or define the transporters as being active or passive. Active transporters require ATP or energy to work. Passive transporters do not. What defines active transport is that it needs ATP to work, and why this is the case can vary considerably. The simple example is a transporter that needs to change its shape. ATP provides the energy for this transformation to occur, allowing it to either open or close. In other cases, it's because the molecules being moved go against the gradient. That is, your cell moves materials from a low concentration to a high concentration, and this is effectively going against nature, in a very simplistic term. And that's because most things will diffuse in other words, move from high concentration to low concentration. But if you want to go the other way, it's not easy to do, and so you require ATP. There's also when you want to move particular things. As a general rule, you can lump your transporters into three kinds of groups. You have a uniporter, or a one molecule in one direction transporters, symporters, which bring in two different molecules together and nearly always in the same direction, and the last is an antiporter, wherein one molecule enters and one leaves. In other cases, your active transport may require a carrier molecule, and in this case, you can think of the ATP as a fuel to power the vehicle that carries the molecule. The passive transporter material is somewhat more simple. There are permanent protein channels in the cell membrane. These channels act like pipes to the outside world, wherein material can move if it is the right thing. The simple example is sodium. Sodium can move around until it reaches an equilibrium. This is possible as it can move through these channels. Now, there are active and passive channels for sodium, so sometimes it will be moved through passive channels, which allow for the equilibrium to be reached, or the active transport in the membrane of neurons, wherein in order to get an action potential, or the neuron signal to travel, you need active transport. There are also carrier proteins. These are typically for a single thing, and these are what allow for a very selective permeability of the membrane. A selective permeability is essentially those three elements of size, charge, and lipid solubility. It ensures that only certain things can get into or out of the cell itself. The reason why you need this selectivity is that if anything and everything can move in and out of cells as needed and wanted, you'd eventually get to a point where there was an equilibrium in what is in the extracellular fluid, and for that matter in blood, and what is inside the cell, including waste material, which is rather undesirable. The other role of proteins, other than being as a carrier or channel, in the phospholipid bilayer is, well, specifically as a structural element. You'll find that proteins are found throughout the inside and outside parts of the membrane. These are attached either to integral proteins, or those channels that go all the way through, or to the phospholipid themselves. The integral and peripheral proteins, that is, those that act as channels and those that are there either on the inside or outside but only on the surface, can do a whole bunch of different things. Sometimes they are enzymes. Other times they act as attachment points. Part of the time they're there as the cytoskeleton, and sometimes they have a role as the recognition or antigen sites on a cell. The second last element of the cell membrane is cholesterol. Cholesterol is effectively a lipid, but in this case, rather than being the lipid tails, this is a completely different element, and it sits between the parts of the phosphate heads and the lipids. It's a much larger molecule. What makes it rather important is that cholesterol in this case acts as a sort of the flexible element, where proteins give rigidity and strength to an extent. Cholesterol provides flexibility, and so it allows for the membrane to be able to shift and move. It means that cells to an extent are malleable, rather than being a rigid solid structure. Further, it's somewhat more responsive to temperature changes, allowing the cell to better cope with those changes. The final element of the cell membrane are your carbohydrates. Now, carbohydrates are somewhat distinct from the phospholipid bilayer itself in that they do perform separately to the phospholipids, but they are very important in what they are doing in there. First of all, the major thing they're going to do is act as an antigen site. This means that they can allow the cell to be recognized as either your self cell, or in the case of, say, bacteria, a non-self cell for the immune system to attack. This is where those earlier mentioned proteins are particularly important. The proteins provide a site for the carbohydrates to attach to the cell properly, and this forms something called a glycoprotein. In other cases, you may find them attached to the lipids themselves, in which case they become a glycolipid. The carbohydrates, when they are part of the cell membrane, tend to be a relatively short structure, somewhere between 2 and 60 units, and they're nearly always made up of a monosaccharide, so think of something like glucose. However, unlike what you might think of with just a long chain of these like a regular metal chain, these can be a little more complicated, as they can either be at that straight linear length, or occasionally they can branch and become more like a tree, and this is more akin to what you would think of with something like starch. These carbohydrates are particularly important, not so much for the human membrane, but particularly for bacteria, let's say, wherein certain carbohydrates are present on bacterial cell walls that are not present on any human cell, and this allows them to be easily recognized as an antigen that tells the human body that this is a foreign cell. By contrast, there are carbohydrates on the human cells that are also used to the same purpose, but unlike the bacterial versions, these are present across multiple species and therefore somewhat more difficult for the purposes of, let's say, immune recognition, but they can be useful for cells to recognize each other and perform their functions. In conjunction with the carbohydrates, there is one other similar sort of structure that's technically speaking not part of the cell membrane in the same context as everything else we've mentioned, and these are receptors. Now, receptors will be covered briefly in this video, but we'll give a full video later on that explains their complete role. In short, receptors tend to be a protein of some structure that has a specific shape to it, and this shape is important as it will only match to a particular signal that comes into it. Now, this can be something like a hormone, it could be a drug, it could be a neurotransmitter. There are all kinds of things that can fit to it. But what is important here is that when the receptor is triggered, it can then create either a cascade directly by interfering with the channels that are next to it, or it can act as a secondary messenger wherein it receives a signal, then there's another signal transmitted elsewhere, and that then tells the cell what to do. Just be aware that these are somewhat more distinct, but what is important is that they come back to interfere with and interact with both active and passive transport. This overall is what your cell membrane is made of, not just why, but how, and to a certain extent why your cells don't simply collapse and fall to pieces at the slightest breeze. They are very much selective in what they allow to pass through, and the passage is based on 3 things: size, charge, and lipid solubility. But there are exceptions based on the various channels that are throughout the cell membrane. Some of these channels are permanently open and will always allow for movement. Others will open and close as required at different times, and then there are those that need to be powered to be able to either open, close, or do something else entirely, depending on the circumstances under which they're being operated. Overall, this again is why your cells won't fall to bits at the slightest breeze, but why at the same time things are not necessarily as complicated as might seem at first glance. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.